Hey guys, welcome back to Computational Biology with Python. In this last sequence alignment video, we're going to explore aligning to the reverse strand or to alternative strands of sequence, right? So you have the forward and the reverse strand. The forward strand is generally what we aligned to, but what if you want to align something to the reverse strand or the sequence that's going to be turned into RNA? So let's look into how we do this. So we'll start from bio-seq, import reverse complement. Okay, so our target for this is going, oops, hit enter a little too fast there. Our target is going to be AAACCC. Our query is going to be AACC. We're going to call our aligner is a pairwise alignment. Let's make this a little bigger for you guys to see. Just see here. Aligner, our mismatch score is going to be -1. Let's put it in here. For some reason this hasn't been working so well. Let's do essentially equals -1, and we're going to say internal gap score equals -1. There we go. Let's just delete this. Okay, aligner dot score target query. So now if we score it, because we have our mismatch score and our aligner score, we get a score of 4. But if we try to align it to the reverse complement, target reverse complement of our query, we get a score of 0. Now what if we do aligner score target reverse complement query, but we say to align to the negative strand. Then we get a 4 there as well. Let's do target reverse complement. Oh, we already did that one. We have to do reverse complement target query. So the default strand is the positive strand. We did target reverse complement query strand negative. Target, let's just do target query. And then our strand, strand equals negative. So just the negative strand. We get a 0 as well. And then, let's see, and that's all 4. So if we align to our positive strand, we get a 4. If we align to the reverse complement, we get a 0. If we align to the negative strand, which is the reverse complement, we get 0. And if we align to the reverse complement of the negative strand, which is like a double negative, which equals a positive, we get the positive strand, which is a 4, right? So that's how you can kind of manipulate that, the reverse complement and the leading or the positive strand. Alignments equals aligner.align target query. So we've saved these alignments into alignment now, so we can say the length of alignments is going to be 1, which means that there's 2, right? So print alignments 0, which is the first alignment, and we can see that here is our alignment for between our query and our target. We could do print alignments 1. Do we have, wait. Oh, no. Okay, so when we say length, it's going to tell us the length of the number of objects, but the numbering is 0. So there's only 0 in the alignment. If that makes sense. I hope it does. Let's do alignments 0. And we can save this to a different file format. So some of these alignment, we have like bed files is a type of alignment format that you can save to. And it will give you the specialized like bed files and what are some of the other ones? PSL files, we don't use those very much. SAM files. Those are all alignment files that you'll get, especially if you do like sequence alignment things. These are the type of files that you'll receive from doing these big global like genome RNA-seq alignments and things of that nature. So you can save these alignments to those type of files. Let's see. And what did we just print? Alignment format bed. I've lost my spot because I got a tangent about bed files and SAM files. We can also do alignments equals aligner dot align target reverse complement query strand equals negative, and now we could do print alignments format bed formate so you can see that we get the same for if we do the reverse complement of the negative strands, it's like a double negative again, right? So we get the same thing as our normal alignment in that case. What if we do, I'm just going to copy this and edit it. So what if we just do the negative strand, and then print the alignments. Just copy and paste that too. Alright. List of index out of range. Let's see, is that right? Print our alignments equals aligner target query strand equals negative. Oh, let's do length of alignments. Length of our alignments is 2, which is good. So print alignments 0. You can see here's our alignment, which just shows it's completely unaligned. And then if we do print alignments 1, you can see the same thing. It's just the reverse, the backwards of it. So that's why I didn't like to try to print it out, because there's just no alignment here, right? It's super negative score. There's 0 overlap between these two. Okay, so let's do one quick. What are we at? All right. Aligner left gap score equals. This command hasn't been working, so we might have to. Oh, hey, it worked. Aligner right gap score equals negative 0.2. I don't know why they would be different between left and right, but we're going to do it anyway. target query. So we have an alignment score. Wait a second. Why does it not match what I did before? Negative 0.5. Okay. All right. Well, let's keep going on. Alignments equals aligner.align target query, and then we'll do length of alignments. We get a length of alignments 1, so that matches still. So print alignments are only alignment, and we get AAA CCC versus AACC. Good. Now what if we do our alignment score, we compare it to the reverse complement? Target reverse complement query strand. Oh, what is going on here? Hold on. Alignments. So we're probably going to get the same thing, right? Alignments. Oh no, wait. It's aligner. Hold on. What are we doing here? Alignment equals aligner score target query. Oh, we skipped a part here. Aligner score target reverse complement query strand equals negative. Okay, so we have a 3.3, right? Because that is just again, the double negative, reverse complement of the negative strand. Now, if we print alignments 0 for this, you can see we have these same alignments. So, but all right, let's do one more thing. I'm just gonna paste this down here, and I'm going to make this the positive strand. And then print. Actually, no, we're gonna do something. Don't do that. Do this. We'll make this the positive strand and see what the score is then. So you see the score is -2, right? So that shows that printing or mapping our DNA to the reverse complement gives us a worse alignment than mapping to our standard string. So if I just change this, say query, we should have the 3.3, right? Because it's the positive strand. Yes. So if you do a double negative, so if you do the reverse complement and then map it to the negative strand, it like cancels each other out, right? It's like multiplying two negative numbers. If you do just the reverse complement or just the negative strands, then you're mapping to the opposite strand, and you're getting a poor score in this case. So it depends if you're using, cDNA or RNA or some of these different, I guess I'll say versions of the same DNA, say different transcripts of the same DNA. Then you can manipulate this pairwise alignment to better suit your needs in that regard. So there's a lot of pairwise alignment stuff. I could almost do, like we cut out muscle because it just involved too much installing of packages. So we're just not going to hit that up. There's a lot of pairwise alignment. I just gave you a taste, like a 25% taste of all the different things. I'm still debating if we're going to go and do like Smith-Waterman and some of these actually learn the theory behind the alignments. We'll see where we get with some of the other stuff that I want to do. I don't want to put too much content up, but at least initially, eventually at least on the YouTube channel, I'll get to it. But yeah, that's pairwise alignment. So if you have a piece of DNA, you're trying to match it to the genome of an organism, or seeing if these two genes that you think are closely related are or not, things of that nature. That's what this pairwise alignment is generally used for. You can also, like we saw with Clostal, you can create phylogenetic trees by determining how closely related sequences are by the number of mismatches and gaps and mutations and indels and things like that in there. But yeah, so that's your brief introduction to BioPython. Like I said, if you want to get more into pairwise alignment, there's a ton of stuff, a bunch of different algorithms, a bunch of different packages that you can use. Like I said, it could almost be a whole course, and I'm not going to subjugate you to that. So if you're watching on YouTube, please hit like, please subscribe. Thanks for watching. I hope you learned something. It was very cursory, but you get the idea of how to do pairwise alignment now. If you want to dive more into it, check out those help videos, or BioStars or BioPython or all these different resources. So thanks again, and I'll catch you in the next one.